Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. So the good news is this week we're wrapping up chapter 10. And a little bit more good news is that we're doing this by talking about the genetics of viruses and bacteria. Not sure if that's good news for you, but for me it's good news. So I've decided it's good news. Let's get into this. As always, right now, my terms and definition slides should be popping up around me so that you can grab the vocabulary we will be using today. However, as always, I'm going to give you a quick reminder that if you would like the entire PDF version of my slideshow, you can head on over to my Patreon. The link is down in the description below, and you can take advantage of the PDF slideshow and any other study material that I have decided to come up with to help you guys out in your journey to learn biology for absolutely free. However, if becoming a patron is something that interests you, you will get all the study material and these videos at least a day in advance, give or take a few hours. So what exactly is a virus? Well, a virus is basically this little particle that can infect living cells, and it consists of genetic material either in the form of DNA or RNA, and a capsid, which is a protein coat. Some viruses also have like a membrane envelope around that protein coat, but we will talk about that a little bit more in a couple of slides here when we talk specifically about animal and plant viruses. So for now, the big things that are important about viruses and what makes them are that genetic material. So in this diagram here, the little blue squigglies showing DNA, and the capsid or protein coat that is the yellow part. Now you'll notice there are other parts of that protein that make up the tail fiber, so things to help the virus kind of move to its next host cell, but the big deal is that a virus is genetic material surrounded by protein. And viruses are also not generally considered to be living despite having DNA or RNA. And the reason for this is because they don't display all of the characteristics of life. One big one is that they can't really reproduce on their own. They have to use a host cell to reproduce, and not necessarily in the same way that you see a parasite using a host. And we will get into parasites in future videos. Today we're talking about viruses. So let's talk about the life of a particular kind of virus known as a bacteriophage. Now, if you remember a few videos ago when we were talking about the beginning experiments that figured out DNA is what carries genetic information, a bacteriophage is just a very specific virus that attacks bacteria. Bacterio, the bacterio part, phage just stands for virus. And generally, if you're talking about viruses, you're just going to refer to them as a phage if you're talking about the particular thing. So for the life of a phage, there are two possible pathways. One is the lytic cycle, where the phage will inject its genetic material into a particular bacterium. That viral DNA will form a loop. Then the host cell will be used to make more viral parts. The new phages will be built, and then the host cell will burst. The other possible pathway is known as the lysogenic cycle, where the phage will inject material into the bacterium, the viral DNA will form a loop, but then that viral DNA will insert itself into that bacterial chromosome, and the now lysogenic bacterium will start replicating normally. So I know that all sounds a bit confusing, which is why we're going to break down each of these cycles individually. So the first thing that we need to notice about these life cycles is that steps 1 and 2 are the same. So step 1, we can see that that phage attaches to the host cell and injects its DNA or RNA if it's an RNA virus, but for simplicity, we're going to refer to it as DNA. And then the capsid or protein coat is just kind of discarded. The whole job of the capsid is just to protect and get that DNA to the next host cell. So once the genetic information is injected, there's no more use for it, and it's just kind of left to wander. Really, it's just going to end up getting broken down, but that's not important. And then step 2, that viral DNA is going to form a loop. Now, once that DNA forms a loop, it can embark on either of the 2 pathways. So first, let's talk about the pathway that's a little bit more familiar, mainly because it's the one that we talked about a couple videos ago when we talked about those experiments. So that would be the lytic cycle. And so the lytic cycle, after that second step of the DNA forming a loop, goes on to step 3, where the host cell is then used to make viral DNA and proteins. So it just kind of hijacks the inner mechanisms of the cell that can make more DNA and make proteins, because the viral DNA cannot do that by itself. The virus on its own does not have the necessary machinery to make more DNA and to make more proteins. It has to use the machinery of the host cell to reproduce itself. And so once more viral DNA and proteins are made, the new phages are going to assemble inside that host cell. And then once the phages are assembled, the host cell is going to lyse or burst, and the new phages are going to be released, and the cycle can begin again because they're going to go along their merry way looking for another bacterium to then infect. However, there is the second possible cycle that a bacteriophage can then enter, and that's the lysogenic cycle. So the big difference here is after that DNA forms the loop inside the host, instead of going on to hijack the machinery of the bacterium, it's going to insert itself into that bacterial chromosome. And once that happens, the viral DNA that is inserted into that chromosome is known as the prophage. So the phage there indicates that it's viral, and then it's just basically kind of part of the bacterial chromosome at that point. And then the bacterium is known as lysogenic because it has that phage DNA inserted into its own DNA, basically. And once that happens, the bacterium is just going to live out its life. It's going to replicate, it's going to divide, it's going to go through binary fission, just like normal. And this can happen many times before the 5th step occurs, which is just some sort of environmental stress happens. And that environmental stress causes the viral DNA to separate from that bacterial chromosome and form a loop. And then we end up back at step 2. Once we end up at step 2 again, it's possible that this particular bacterium with that viral DNA in it may now enter the lytic cycle. Or, depending on what the environmental stress was and if it's still there or not, it may go right back into the lysogenic cycle. So if we are at step 2, either of these pathways is possible. As we can see here when we put this diagram together, if you enter the lytic cycle, that's it. The pathway is chosen, and the only outcome is that the host cell is then going to burst and new phages are going to be created. However, if you're in the lysogenic cycle, it's kind of indefinite how long you're going to end up there, and the environmental stress will send you back to step 2, but may or may not result in you then entering the lytic stage. So those are the 2 possible pathways that a bacteriophage can embark on in its life. All right, so we talked about bacteriophages. So what about animal and plant viruses? So The typical animal virus is going to have that membranous outer envelope that we briefly mentioned at the beginning of all of this. And one important thing about this outer envelope is that it's going to have these little projecting spikes made of glycoproteins. And what all of this ends up doing is it helps that virus enter and exit cells. So the example we can see here in this diagram is a 2D depiction of the mumps virus and its general life cycle, because this is pretty typical of your animal viruses. And another thing here is that while bacteriophages, we saw DNA, animal viruses tend to have RNA as opposed to DNA. And then also most plant viruses will be very similar to the animal viruses. They do tend to have RNA as well, but plant viruses, once they kind of get past the epidermis, which is that outer layer of the plant, and actually are able to start infecting cells, they travel very easily from cell to cell through the little connections between the plant cells that are actually known as plasmodesmata. However, that term is not really necessary for you to know at this point. Just plant viruses and animal viruses are very similar in that they have that outer envelope that's very membranous, so it's made of a membrane, and it tends to have those projecting glycoprotein spikes. And then the plant viruses, it's a little bit easier for them to travel cell to cell because plant cells have the connections between them. So what does the life cycle of a plant or animal virus look like? Now, for simplicity, we're going to be looking at an animal virus, but plant viruses are similar, just with a few differences, like being able to easily move from cell to cell without necessarily needing to exit the cell in the way that plants do, in the way that animal viruses do. But step one, that viral envelope is going to fuse with the cell membrane. So remember that they're both membranes, so they easily can fuse together. And inside that envelope is the protein-coded genome RNA for that particular virus. So once that fusion happens, the protein-coded genome can then enter the cytoplasm of the host cell. And once in the cytoplasm, enzymes are going to digest that protein coat and free the RNA that carries the viral genome. And so once that protein coat is digested, step 3 is that another enzyme that was actually contained inside the protein coat with that genome is then going to use the RNA as a template to make complementary strands of RNA. Now, the new strands of RNA are going to be used for two things. One is going to serve as the mRNA for protein synthesis, and the other one is going to serve as a template to make new viral genome RNA, which is why it's important to kind of distinguish between which RNA you're talking about when you're talking about the life cycle of an animal virus. So once the protein and the genome RNA has now been synthesized inside the host cell, everything can then be assembled. So the protein coat will then end up around the RNA again, and the virus can then leave the cell. But the sneaky thing it does here is it uses the cell's own membrane to kind of bud off and then be released and go look for a new cell to infect and re-begin the process. So animal viruses can enter and leave the animal cell without necessarily destroying the cell. And that is a big distinction between animal viruses and bacteria viruses. So a few quick notes. Again, the cell may or may not be lysed. In some cases, the cell will burst. However, in some, like the mumps virus that we're looking at here, the cell is not lysed at all. It remains intact and basically remains as a little virus-making factory. And not all animal viruses are going to be replicated in the cytoplasm. There are some viruses that will replicate in the nucleus. One example being herpes viruses, like the chickenpox, cold sores, and shingles viruses. And then we also have viruses that once they enter a cell, may remain dormant until a particular stimulus triggers it to become infectious and begin to cause symptoms. Again, there are kind of exceptions to the rule, but the general life cycle when it is infectious is going to look like what we went over. So this brings us to emerging viruses. Now, emerging viruses, like we see on this map here, are viruses that kind of pop up suddenly and kind of make scientists scramble to figure out how to fight them. Now, note that we are currently, at the time of filming, still in the midst of one emerging virus that is currently causing a global pandemic. That is not on this map right now because the book we're using came out prior to the pandemic and prior to the discovery of this new emerging virus, COVID-19. But other viruses that you may recognize on this map are things like H1N1 that popped up a couple of times throughout history, the West Nile virus, avian flu, Ebola, Zika fever, and AIDS, which is actually caused by the HIV virus, which was redundant because V in HIV stands for virus. This brings us to retroviruses, which are another interesting class of virus that we're going to talk about because that's another kind of modification on the classic viral lifestyle. A retrovirus is an RNA virus that reproduces using DNA. So it basically has to reverse the normal flow of genetic information. Remember that normal is DNA to RNA, but in the case of a retrovirus, we get a flow of information from RNA to DNA. And one example of a retrovirus, which is actually shown in this little cartoon diagram here, is the human immunodeficiency virus, or HIV. So the virus that is responsible for causing the AIDS disease. What exactly happens? How does a retrovirus end up going from RNA to DNA to reproduce? Well, it's going to enter the cell very similar to a normal virus, so we're going to skip those steps. The important thing here is that the retrovirus uses a thing known as RNA transcriptase, which uses RNA as a template to then make DNA. So, whereas RNA polymerase in the normal chain of events uses DNA to make RNA, RNA transcriptase uses RNA to make DNA. And then the kind of second step, once that first DNA strand is made, is the RNA transcriptase then has to make the complementary strand of DNA. And once this kind of double-stranded viral DNA is made, that DNA can then enter the nucleus and insert itself directly into the host chromosomal DNA. And then from there, it's actually the host cell that ends up making more viruses when it goes through the normal processes of transcription and translation and goes over the portion where the viral DNA is in the host chromosome. So it is a very clever system for this virus to then reproduce without having its own DNA, really. It just hijacks the host's own mechanisms by inserting itself into the host chromosome. So again, once it's in that chromosomal DNA, the host's own RNA polymerase will transcribe the viral DNA back into mRNA, and the RNA will be translated into viral proteins, and then the new virus is able to assemble itself and leave the cell. And then the process starts all over again. And so before we wrap up by talking a little bit about bacteria genetics, we're gonna pause here on something known as the prion. We're not gonna get in depth in this. This is just kind of an aside that our book mentions that I don't know if an intro level bio class will really go into, but it is a kind of interesting thing. Now, here we just see a computer model of a prion, but basically a prion is an infectious protein. So it's not a virus, it's not a bacteria, it's just a protein. And specifically, it's an abnormally folded brain protein. And when it enters a cell with normal proteins, it will cause those normal proteins to then misfold. I think that the particular mechanism for this is still not entirely understood. But once those proteins are misfolded, they will clump together and they can disrupt brain function. So that's as deep as we're going to get into it. It's just a little interesting aside in the book, and I thought it was interesting enough to keep included in our discussion here, since we are talking about things that can be infectious. So this brings us to bacteria. So how do bacteria transfer DNA? Since we know that bacteria are single-cell organisms that primarily reproduce asexually through binary fission. Well, there are three main ways that the transfer of DNA can happen in bacteria. The first one is called transformation, where the bacteria are literally just going to pick up some genetic information from their environment. So that DNA fragment from perhaps another bacteria that has been destroyed is kind of floating around. So before it's degraded too much, the bacteria of interest will pick it up and then just kind of get that brand new piece of DNA, and it'll somehow insert into that bacterial chromosome. So transformation, it picks up new genetic material from the environment. Another way is through transduction, which is where a fragment of DNA from another bacterial cell is transferred into the bacterial cell of interest through a phage. So basically this DNA had to have come from a former host of this particular phage, and now it's been transferred into a new bacteria, adding new genetic information. And it's through this viral transfer of DNA that we do a lot of experiments in genetics now. But I digress there. The other form of DNA transfer in bacteria is called conjugation, and it's basically bacteria sex or bacteria mating. So a mating bridge will form, the donor cell will pass over some DNA, and then the recipient cell will end up with that DNA. There can be crossing over that will occur, or that new DNA can just be entirely incorporated into the new bacteria's genome. But let's take a little bit of a closer look at conjugation, because it does get a little tricky and it does require something specific. It requires something known as an F factor. And an F factor is basically a piece of DNA that carries the genes to make what are known as sex pili. And the sex pili are required to kind of trigger that mating bridge to form and to make conjugation happen between two bacterial cells. And the F factor can exist either integrated into the main DNA chromosome, or it can exist as a plasmid, which is basically just another little piece of DNA looped inside a bacteria that's not connected to that main bacterial chromosome. In the case where the F factor is integrated into the main bacterial chromosome, recombination can then occur in the recipient bacterium, and like crossing over can occur. But if the F factor does exist as a plasmid, then the genetic material that is shared via the mating bridge will also become a plasmid in the recipient cell, and then that recipient cell can now act as a donor to other bacteria in the future. All right, so that's all I really have for you guys today. I do hope you have a wonderful rest of your day, afternoon, evening, what have you, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane B. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.